Well, hi everyone, and welcome into Pushing the Limits. Today I am super excited. I've been so excited about this interview for so many weeks. I'm just absolutely ecstatic to have Dr. David Furman with me. So welcome to the show, Dr. Furman. It's absolutely wonderful to have you. Very excited to meet you. Dr. Furman, you are the director of the 1000 Immunomes Project at Stanford University and associate professor at the Buck Institute, also co-founder of Edifice Health and Cosmica Biosciences. And today we're going to be doing a deep dive into your research and the wonderful work that you've been doing over the past really 15 plus years. And perhaps we can start though with a little bit of background as to how you got into this area and this arena. You could have done anything with your amazing capacity to understand a lot of complex things. How did you actually end up in the aging space, if you like, and at the Buck Institute and Stanford doing what you're doing?
Yeah, so I was doing my PhD in human immunotherapy and did a lot of monitoring of the immune system in cancer patients. And a professor at Stanford at that time who really, I think we all believe he's going to get the Nobel Prize for the discovery of the receptors on T cells, Mark Davis, recruited me. And I was also doing part of my med school years. And so I dropped out med school, have finished my PhD and moved to Stanford to really try to reinvent the way we study immunology. And so for probably 150 years, we've been using animal models, and for the most part, these don't translate into human biology. And so we decided to focus on human studies, to do more of that type of research. And so when we were doing the 1000 Immunomes Project that we started in 2007, really, so a while ago, that I'm going to allude to in a moment, the one thing that we found repeatedly over and over were aging signals. Yeah. We're looking at immune system, and then we're seeing aging signals, and that turned my entire research to aging. And I thought we should use the human immune system to understand aging. There's a very clear connection here.
And Dr. Davis is really a luminary in the space, so to get an invitation from him to come and work in this space must have been quite amazing, and his work that he's done on the T cell receptors is I expect the Nobel Prize too to be forthcoming at some point. And I remember in one of your interviews that I listened to, you said to your father one day that you wanted to have a massive impact on human health and to have a big impact in the world. And you were thinking about med school, and you went to med school for a few years. But your dad gave you some really sage advice. Right. What was that advice?
Yeah, well, the advice was really that when you treat patients, you do it in a piecemeal fashion, one by one. And you can create impact, but not to the extent that you're interested in by what you're communicating to me now. It seems like you want to have more impact in the humanity as a whole, as opposed to thousands of people. In the best case scenario, as being a doctor, those real doctors that do medical work, right? But in my case, with a PhD, then I thought, okay, this could expand the avenues and really have a massive impact on things that all medical doctors could use. To help more people and have more impact.
Absolutely. I think your dad was very wise to send you in that direction, if you like. And okay, now let's focus in a little bit on the 1000 Immunomes Project and immunology. And I've been fascinated in the last couple of years with, in my studies of the hallmarks of aging, I keep coming across, inflammation, immune system dysregulation, thymic involution, all of these things that happen as we age. And of course, my audience know of my battle with my mum to help her with all her comorbidities that she has, and struggling with her immune system, and especially going through cancer treatments and so on and so forth. And so it really came apparent to me, and when I discovered the 1000 Immunomes Project and started reading your research, I'm like, yeah, this is it, because the immune system is the key foundation of slowing the aging process. Like the paper that you put out, I think it was in 2023, about chronic inflammation is the cause or at the base of all the hallmarks of aging. And that really rung true with me as being like, these are affecting all things. So let's start with the immune system first. How did you sort of, like, were you seeing age-related markers in when you were studying immunology? How did you sort of, come to that?
Right. So, really, this idea that the immune system and certain aspects of the immune reaction, like inflammation, can drive aging processes is rather new, right? In terms of the history of immunology, we've known that the immune system and the inflammatory response will help you clear viruses and infections, other sorts, and if it goes awry, you develop autoimmunity. We've known that for many, many decades, but only in the early 2000s, we started looking at some aspects of immunology that were somewhat accelerating biological aging in different systems, right? And so then the idea of studying the immune system to look for determinants of aging processes and mechanisms, and hopefully develop different interventions to target the immune system and extend health span and lifespan by default. Was kind of the focuses of the of the 1000 immunomes that
you sort of alluded to something before, which was that all the immune system science to date has been mostly in animal studies, and the animal studies just really don't translate, and that we've got this sort of a problem. It takes 10 plus years to develop a drug, $2 billion or something to bring a drug to market. 90% of our drugs fail. We're just not getting, where we need to be with that model. And so you've decided to focus then onto to actually studying human immune systems. That's quite a shift, because usually we start with mice or C. elegans or something like that, and then move to mice and then move to humans. Why is your approach, like to go straight to working on humans, really quite revolutionary?
Yeah. This is really something I didn't start. It was really a trend in the field to better model the human biology. We have been in this revolution of technologies that now we have to extensively monitor every single aspect of human biology, from all the genes that are in the genome, all the proteins that are in the proteome, all the metabolites. We're talking about hundreds of thousands of molecules from blood. So it is really a good time for us to start thinking about, monetizing, if you will, on those technologies to, or capitalize to really understand the immune system in humans. As you alluded to, there's almost no good model for human disease, let alone human aging. I mean, it's really, really poor models. All the animal models that we see, from C. elegans to all the way to animals like mice, or even non-human primates is probably the closest that you can get. But we're using humans, and we're using human organoids that are mini, veins or mini hearts or mini immune systems. We develop in the lab to really have the genetic and the phenotypic variation that we see in human beings, and not be looking at the wrong place. Although, we've used animals to really dig into the mechanism, because oftentimes with human studies, it is challenging to understand mechanistic behaviors. But for the most part, those even those do not translate as well. So I decided to really focus on human biology, biopsies, blood, and large studies, and measure everything under the sun, and then try and use AI machine learning to cope with the data and get significant insights into
because
the
this is
biology
a
and health
convergence of
disease.
technologies, haven't we? We've got computational, sciences, and we've got all these exponential technologies all happening basically at the same time in history, which gives us the opportunity now, like never before, to be able to do these things. Like you couldn't do the computations that would have been required previously because you just wouldn't have had the manpower to do that. And now, with computers, they can do things in seconds what would have taken days. So this has accelerated the rate that we can research. Or decades.
Oh, well, it would have taken decades.
Wow.
Yeah, well, it would have taken decades, honestly. So the power to store, this is not minor, and analyze and compute, now is basically unprecedented. And anything that you want, you can do today. We're using these supermassive language models and to, extract relevant information from 30 million papers in
That's amazing.
half an hour in the time my lunch during my lunchtime.
You must be like a kid in a candy store sometimes when you think about. I mean, I know even with my basic knowledge of very basic things, and I can do things that I couldn't even do 3 or 4 years ago. Just with ChatGPT, let alone the stuff that you guys have available to you. It's so. This is really going to revolutionise. I mean, I really hope that within 5 years' time, 5 to 10 years' time, we're going to be sitting in a completely different world with completely different paradigm of the way things are done. My fear is that the clinics aren't keeping up. Like we're already not. We're already I dare say 20 years behind just in New Zealand where the current research is, and I came across this when I was working with my mum with cancer and so on, and realizing there was all this information that I was getting and learning, and none of it was here available, and from a technological perspective, and also none of it was available even when from the clinicians' perspective, who are all overwhelmed, overworked. It's impossible for them to all keep up perhaps, and there's protocols and ways that we have to do things, and it's like moving a cruise liner. It's really a tough shift, but if this is why I'm passionate about this podcast, because it brings from the cutting edge, people at the cutting edge like you are, to the consumer to hear what is actually happening out there, to put some influence perhaps into the clinics to actually create action, to get the doctors to actually be doing some of the latest and greatest things that are out there and that are available. I think it's really, really key.
So there are several buckets. I'd like to break that down into different buckets. One is the regulatory bucket. FDA and organizations like that one are there to protect us. So if it's going to take too long, it's a slippery slope as well, right? So to the extent that at some point they're somehow delaying something that could be implemented prior, then it's a slippery slope, right? But they're there to protect us, and they're doing a good job on that regard. Now, as far as the technologies go and the incorporation into the clinic, I mean, on the one hand, it's a slow process because there's no payers, right? So this is mostly coming out of pocket. And so it's a very slow adoption of these different technologies to assess preventative care in the beginning, in the very basic of, like, you're measuring inflammation and what are the markers out there, or you're measuring biological aging, and all of a sudden you're paying, $2,000 for worth of different analyses there, and that's all out of pocket. So there's different buckets there. It's obviously the one that relates to the adoption of different technologies that could be maybe not that expensive, and that different governmental agencies and healthcare systems will have to adopt. And the reason is they're behind,
Yeah.
and we know they're behind, right? But there's so much pressure out there from clinical and private parties, right, that now are taking over. I mean, the best clinics probably in the world today are private. And so
Yeah.
there's a whole shift there, and adoption of technologies is definitely a big theme.
Yeah, no, that is so true. There are many obstacles for us to overcome, but it is really exciting that we do have now some of these technologies. And let's dive into a little bit into the 1000 Immunomes Project and walk people through a little bit. You took 1,000 people basically 15 something years ago and took blood tests. Basically, Stanford has a wonderful hospital. You were able to get access to blood from 1,000 people. Can you walk us through the 1000 Immunomes Project, how it was set up and what it was targeting? So what was the outcomes of really of that? Because that's just super exciting.
Yeah, thank you. The basic idea was to create the first multi-omics, and we didn't call at that time multi-omics. That's a newer term. But like a systems biology approach where we're looking at a variety of different biological features from blood and from responses to vaccination, and to study this in a large cohort of people, let's say 18 to 100, and to try to understand that at the cross-sectional level. So that means once, in the timeline, or longitudinally. And so, for example, around 150 of those individuals were followed longitudinally for now 14, 15 years, right? So there's a lot of insight into the trajectories of aging. So you not only can study the population behavior as to things that go down or increase or may be beneficial or even detrimental, but now you can see at the subject level how they vary. So an individual would have an accelerated aging based compared to the population, or it could be more protected. And so you're learning from those outliers that were identified in a way. So the idea was overall to start a technology center, and we did so. It's the Human Immune Monitoring Center that was partly funded by Larry Ellison in the Ellison Foundation, and a lot of also federal grants that helped to put together this one-stop shop for immune monitoring and biological monitoring in these individuals. And that got pretty quickly adopted as a model and by other faculty members at Stanford and also outside of Stanford, where they started to create these facilities of different technologies that can monitor the biological changes that occur in diseases, or just longitudinally looking at it.
When you say you take people's blood, what are you actually measuring in the blood? Is it thousands and thousands of molecules and proteins? Yeah,
right. So from blood, basically fractions in blood that everybody knows from the textbook are cells, and the soluble fraction, which is plasma or serum. And so we use that material to study gene expression changes in the immune cells. So it means the entire genome and the expression of those genes we would measure in all 1,000 individuals. We also measure the frequency of immune cell populations by using a variety of different tools. Now we're looking at over 200 different populations from blood. And then we do proteomics that studies basically a whole proteome, almost a whole proteome with 10,000 proteins, for example. When we started in 2007, I must disclose, proteomics was only 50 proteins, right? A very targeted kit that you would use, but the premise was exactly the same. You would study as much as you can using state-of-the-art technology, and then try and derive mechanisms and interventions
Yeah.
for
And
age-related
you've
pathology.
come out with some really key proteins, and like you've developed the iAge clock, or the inflammation aging clock, and we know, like, most of my listeners have heard of other clocks, the Grim age, the Glycan age clock, True Diagnostic has epigenetic clocks, and so on and so forth. And this is another clock of aging, the inflammation age clock. Can you tell us, this is a little bit different though. Is this measuring proteins in the blood as opposed to, say, DNA methylation markers, which, like I've used them somewhat in the clinic, and I've used them for myself, done some of the testing. Haven't found it to be too conclusive yet. Like what's the actionable takeaways from it? I don't feel like we're quite there yet. Like we need more time perhaps for the epigenetic clock. So why is the inflammation age clock different, the iAge clock, different than those other clocks, so to speak?
Yeah. So the main difference is that you're looking at soluble proteins in the plasma or in serum, right? And whereas the epigenetic clocks are looking at methylation patterns in the DNA that are still elusive as far as the functional effects of those CPG islands of methylation sites in the DNA that we know change gene expression, but what do they mean exactly, and how they relate to different exposome factors or lifestyle decisions, or we're starting to know a little bit better, but it's still very elusive. And so, yes, you would see a very accurate clock. That means that, close to the chronological age, and deviations may inform about diseases at some point in terms of the studies that have been published. But then when it comes to interpretation, what do you do with that is I think still something we don't know how to interpret quite well. When you have the proteins in the blood, that's much more straightforward to interpret. And there are things that you can do to upregulate or downregulate expression of those proteins. So say protein A is detrimental for cell senescence or for cardiovascular aging and so on. You could take action and try and download or downregulate the expression of these proteins. Whereas in the methylation, how do you change methylation patterns?
Yeah, that's a really good thing, because when we look at the iAge clock, and you've got some pretty key markers there. I mean, you're measuring an awful lot in the background, but in the inflammation age clock, you've got just a handful of markers that we can really, from a clinical perspective, I'm quite excited and hoping that this will be available worldwide when you do launch, and not just in America. Yeah. Is that going to be possible with the Well, that's exciting.
Yeah. Oh yeah. No, absolutely. We are establishing at the moment, actually, a very interesting question, because we're establishing partners in Europe. We have partners in Latin America, and there's a group in Prague as well. That is a quite interesting part of the European deal that we're negotiating at the moment. And also potentially partners in China.
Okay.
So
because I'm hoping, because I'm in New Zealand, obviously, and we get nothing down here much. I'm hoping that this will be available worldwide, because in a clinical setting, like if I can use this clock to help my clients, see where they are before we try an intervention, and where we are, say, 6 months later after doing whatever interventions we're doing, are we moving the needle? That would be a very powerful.
Yeah. I think the idea of actionable results is key here. I'm going to give you a very simple example. CXCL9 protein circulating in blood is associated with elevated arterial stiffening and left ventricular hypertrophy, and not really good endpoints as far as the cardiovascular aging goes. We also know that it increases with exposure to formaldehyde. And we know that formaldehyde can be everywhere where you have new furniture, for example, things like that. And so you can start measuring more formaldehyde in the air, and that gives you a very directive way of approaching your client or your patient.
That's very clear. And the CXCL9 is something that I've, because when I was looking at the markers, you've got trail, you've got CXCL9, you've got eotaxin, you've got MMP9, I think it was, you've got a number of other ones as well. But I don't know if the MMP was in there or not, but it's one of the things you measure. Thank you.
It's part of the gene expression, markers.
I hadn't heard of a lot of these, like we tend to look at things like IL-6 and, TNF alpha and NF kappa B perhaps, and interleukin 1 beta and things like that. But these ones I'd never come across before, and I was fascinated. And the CXCL9 being one that seems to be quite a major player with this cardiovascular aging, if you like, and just trying to research for my own thing what can help with CXCL9, and I wasn't aware of the formaldehyde one. So, but this is where the power lies. If we can get these markers, these targets that we're wanting to change, we're wanting to lower CXCL9, we're wanting to lower eotaxin, if we can measure them, and then we can try different interventions. And I know that you're working on that, what interventions. But yeah, tell us a little bit about those markers perhaps, and then where you're at with the current interventions that we can try. I think you've done one with quercetin, which was an interesting one.
So let me first discuss the technology aspect of it, and then the implementation, because these are different things. So on the technology side, we can take those markers and run what we call gene compound enrichment analysis that basically uses the entire interactome between genes and chemicals. And that can give you a list of very positive regulators and negative regulators of each one of the proteins. And these could be things that are in the air or in your foods, or either positive or negative. So you can start mapping the different compounds to foods and food servings, and so recommendations for supplements and stuff like that. So I think in terms of the technology, we can even nail down to the dosages that you may need as in daily dose to up-regulate or down-regulate the expression of that protein, right? Now, whether or not we as a company, Edifice Health, the company that is commercializing this test for inflammation, is going to give the end consumer all this information, maybe not. Maybe that will be for their doctor, and will have the responsibility to discuss that with their patient. But as far as technology exists, implementation may be through
that's powerful, like even for being able to test. So I have a biotech company, we're doing going after immunosenescence is one of our formulations that we have. I would love to be able to use that test to see, are we moving the needle? Is there possibilities of perhaps doing even clinical trials with different partners, so that we can start to see some of these things that we think may affect these, and then actually run different trials to see whether we actually are going to move the needle on these things, because then we've got actionable things that we can actually take away and we can show. Because when I'm working with clients, if I I can give them a protocol for what I think is the best supplements for their particular situation, perhaps, right? But I'm unable to measure a lot of it, and people often, when they're taking a supplement, expect it to be like taking a Red Bull. They're going to feel more energy tomorrow, and it doesn't work like that.
So let me just give more flavor to that comment, there's been a need in the aging field
Yes.
to identify biomarkers of aging that are reliable, that can be used as a surrogate markers for aging, that you don't need to wait, 15 years until you see a change. So these biomarkers that are informative for interventions, especially for interventions, are needed in this space. Now, you probably know the X prize on healthspan. That is $101 million. We're applying on two different hats I'm wearing. One is Buck Institute, and the other one is the study in Toulouse in France with Professor Bruno Vella. And it's very clear that they need endpoints to assess the efficacy of the aging interventions, or the different things that they're requiring to just to remind everybody in the audience, and maybe to put that to discuss that for a moment. This is a study that should last for 1 year, so it's 1 year intervention, and they're expecting to see a 10-year improvement in functional aging. Okay, and with a target of 20 years. So it's very, $101 million for the price. And so, but they want these different endpoints that you need to have. We're currently in discussions with the leadership at the X Prize to incorporate inflammatory age as an immune endpoint to assess the efficacy of all the interventions for all the different applicants
that's
to the X Prize.
So this is how powerful this test is. This is really
it's getting to,
because I'm very
much
much into prevention and having lived with a disabled mum now for 10 years and having seen the stress that it puts on the family, the financial costs, the hell basically that you go through when you are dealing with the aftermath of some major ailment. If there's anything I can do to help people be in a preventative mindset, but it's a really hard sell. It's like trying to sell insurance, right? Mm-hmm. You're like, how do you measure it? How do you measure prevention? How do I you just maybe don't die, but you don't know that, it's really hard to get people motivated when you have no endpoint, when you have no measurement stick, so to speak. And that's why I'm excited about this.
It's what, yes, it's a biological feedback, if you will, a measurement that is very actionable, accurate, and informative, and it's associated with a lot of clinical parameters.
Exactly, so because when we go and look at things like CRP or interleukin-6, they're more of acute inflammation, and actually we should define what inflammation is, because we use this word inflammation, but actually there's I heard you say in one of your interviews, you don't even like calling it inflammation because it's very different than the inflammation that we have the picture of in our heads of maybe twisting your ankle and it's red and it's swollen and it's hot, and that's inflammation, right? But we're talking about systemic chronic inflammation here that we're measuring. Can you just give us a bit of a difference between acute inflammation and sterile inflammation and what that is?
Yeah. Right, definitely. So what we know from the textbook on redness, and it gets warm and it's swollen, those are cardinal signs of acute inflammation after you've suffered from an infection or cellular stress, right? That's acute inflammation. There are well-established markers. Some of them you alluded to, interleukin-6, CRP. So currently, the most savvy cardiologists out there are using CRP to detect, suspected infections.
No,
they're not using it for cardiovascular disease anymore.
Yeah. It's random.
It's not strong. It's not accurate enough to detect early on cardiovascular events in the future. So I think there's a need for better markers there. Of chronic inflammation. Acute inflammation is very well defined, as I mentioned, right? And there are well-established markers. For chronic inflammation, and it's age-related, this is like fighting a ghost, right? Because in a way, there's no such thing as an infection. When you're raising your inflammatory levels as you get older, it's largely due to the environment, to the way we treat our
Yep.
bodies, the pollution in the air, the quality of the water, the presence of plasticizers and plastics in the food, and all sorts of hyperglycemic foods that's highly inflammatory, having a poor microbiome diversity and richness of the microbiome is also associated with high inflammation. And so it's basically the environment is fighting a ghost. I like to say that's why,
Yeah.
measuring is so key, but calling it inflammation, it may be
Yeah, and that's why I like the term the
counterintuitive.
sterile inflammation. It doesn't come from infection. And it's really the question, which is the chicken and which is the egg scenario. Like, does the inflammation cause the aging, or the aging cause the inflammation? And it's sort of a both would probably be the answer.
Yeah, it is both. And we have data to show that it's both. Just with aging and no exposome factors. So say a sterile culture of organoids, you still see signs of aging that are expected to see in a living organism. Okay. Now, we also know that when those aspects of aging, like inflammation, start to increase with aging, but just a natural aging, those are causing accelerated aging as well. So the more that increases, it's a vicious circle. Exactly. It starts with aging. Just because you're breathing, you're aging, right? And then you produce inflammatory biomarkers that pushes senescent cells to produce more, and that activates a whole inflammatory cascade that basically doesn't stop and doesn't get resolved. So this other idea is like, can we push and maybe boost the resolution of inflammation? The resolution of inflammation is not a inactive process. It's a very active process that requires a lot of energy, a lot of ITP. It comes largely from polyunsaturated fatty acids of the long chain from animal sources, omega-3, call it.
Big fan.
EPA and DHA, those mediate the resolution of the inflammatory response. And again, if you don't have, and most of us are deficient.
I just interviewed someone on that, exactly on that topic, Dr. Michael Lewis, who's an expert on fish oil.
But that basically, results in a reduced resolution phase of the inflammatory response and sustained inflammation.
And I've worked with lots of people with brain injuries, and we have the initial injury where you have the tearing of the axons, or the actual physical injury, but then you have the biochemical cascades that happen in the days and the weeks even afterwards. And I've gone through it with my mum, of course, who had the aneurysm and stroke. It's that's actually, if we can stop the inflammation as fast as possible, lower that. I interviewed Dr. Michael Lewis, who's a fish oil expert, last week, and I've interviewed Dr. Dayan Goodenow, who's a plasmalogens expert, which is another type of fatty in the membranes of the cells that help with this as well. And there are a number of other things. I've interviewed Dr. Barry Tan, who's a tocotrienols or vitamin E scientist, and all these things can help reduce the inflammation in the brain or in the body. And all of these things are going to be beneficial if we can lower that inflammatory cascade, or after COVID, or after a virus, or after whatever the insult is. Yeah. If we can get the body back to homeostasis afterwards and get that inflammatory load down. But that becomes more and more difficult as we age, and we have more from the baseline, more of the sterile inflammation, and then less of an acute inflammatory, which is counterintuitive. Not but it isn't. Well, but it is. You know what I mean? Yeah.
Yeah. Totally. So it's interesting because on the one hand, the resolution of the inflammatory response depends on a sufficient amount of omega-3 fatty acids in circulation, but also if you're consuming high amounts of omega-6 fatty acids that are empowering the inflammatory response, then even if you're taking your 2 grams of omega-3 that you require a day,
No.
you may not be in balance. And so there's also a reduction and cutting on all the sources of omega-6 that you could, possibly do, as in the nutrition side of things, right? So if you can cut on sources of omega-6, and many of those are kind of counterintuitive, right? Like nuts and eggs and avocados, so things that we believe are super healthy, but they're also rich in omega-6, and we should try to maintain low levels.
Yeah, because most of us are around the between 18 and 25 to 1, the omega-3 to omega-6 ratio. And we should be 1.5 to 1, or
exactly.
So
more.
I'm actually like doing high dose protocols with fish oils, with 9 grams a day and things like that, to really try to saturate. If I'm working with someone with brain injuries, for example, that we, the protocols that are really high
Super
dose,
good.
as well as other things like fatty 15 and plasmalogens and other things that I use, but that are all in that same sort of class, if you like. And this is just one, but this is a powerful intervention, right? And this is the thing with this iAge clock, that if we can measure this in a really, quick fashion, and do this successively over a period of months, and we'll get much more people, I think, committing to a protocol, because they can actually see it. Like the moment I can take my fish oil, I don't feel any different, right? I didn't feel anything different from, last month when I wasn't, or whatever. And so it's a hard sell to get people to stay the course, because they can't see the progress that's actually happening on a cellular level, and everything's actually happening on that cellular level, if you like.
Well, that's exactly the place that we want to target. When it comes to tissue and organ level, it's too late, right? So we want to target things where they're early on, exactly as you're saying, at the cellular and molecular level, but you don't see the differences if you don't measure them. So you're absolutely right. It's true. To ensure patient compliance, I think it's absolutely key to see results and to be able to study them as much as possible with different biological aging markers. Inflammatory age is one that obviously we believe, based on all the research we've done, that it's a good and reliable and key marker there. But there are other markers as well. I mean, for example, heart rate variability measured by a wearable device. It's a pretty good, marker of risk for cardiovascular conditions, for example. And that may be indicative and somewhat actionable, right? And but again, I think the fact that we have proteins in blood is very powerful. We can relate that to foods, exposures, and so on, and we can even move the needle. I mean, we've done quite a few clinical studies now, and testing different interventions, and we have a reduction in inflammatory age in some of the studies in a double-blinded placebo-controlled trial that we published in the Nutrients Journal in 200 individuals. We see a significant decrease in inflammatory age. So it is
really
actionable.
Exciting. So when is this clock coming to market commercially, and when can I get it for my clinic? How far away are you from actually having this available, and for clinical trials perhaps too, if we wanted to be involved?
Yeah. So for in the US, this is going to be, it's already happening. We're starting to partner with different clinics and longevity doctors and concierge medicine, wellness centers, most the elite healthcare, because this is a out-of-pocket for now test. And we are in the pilot phase. We did a beta and an alpha, and we're planning to expand pretty rapidly during 2025, so next year. And very likely in 2026, we'll be able to expand to other countries. So today, if you want to send me your serum, and you can ship that, no problem.
Oh, wow. Really?
We've been sending samples all over the world.
So that's doable now, really?
Oh, yeah. So we can do it for
but
researchers only at the moment.
Wanted to do a study or something, we could do that.
You as a partner. Yeah. We did it for the more than 1,000 samples with University of Bordeaux and Toulouse in France. So if you send me your serum, we can estimate your inflammatory age.
That's really
today.
exciting, and roll on the other stuff so that we can actually get it in all the clinics around the world, because I do think there's going to move the needle once people understand what it is that you're doing, because it's quite a shift and it's an educational piece, and this is why I think podcasts like this are actually really important to get the message out and the word out, and to slowly start to get clinicians to hopefully take control of this. I wanted to switch gears a little bit and talk about astronauts and space travel, and what you've been doing of late in relation to the accelerated aging that astronauts experience, which I think most people probably don't know. But when you go to microgravity, or you're in space flight and you're up on the space station or wherever, you're going to accelerate the aging of the systems. Can you tell us a little bit about that and Cosmica Biosciences and what you're doing?
Yeah. Right. Yeah. No, that's a significant shift from what we've been discussing, but not so much. Not so much because this whole new program that I started in my lab, and I started also a company based on that, relies on initial conversations I've had with a principal investigator at the Human Research Program in Houston, NASA, who reached out to me because she said, David, all the research that you've done in the immune system and aging seems to be key for the identification of biomarkers to protect our astronauts from accelerated aging. And I didn't know either that we suffer from accelerated aging in space. But NASA and the European Space Agency have identified several risks and hazards associated with spaceflight, and they're all due to microgravity, not so much to radiation, because you're largely protected from radiation in the International Space Station. But they suffer from 5 times increased risk of cardiovascular disease, astronauts do. They suffer from cognitive deterioration and motor skills. They have immune reactivation of viruses, immunosenescence, variety of viruses that we typically control here on Earth. They reactivate in space. And then also musculoskeletal conditions like frailty and loss of bone mass and muscle mass. So it's really an ideal model to study accelerated aging in humans. It's like the natural follow-up and segue from the 1,000 immunomes that we did on Earth.
Yeah.
But on steroids, right? We're accelerating by about 30 times, 30-fold every single aspect of physiology. And so that really kicked off a whole new program that I started in my lab of space medicine. And so with that, we envision a way of simulating microgravity in the lab. And there are instruments that were developed by NASA engineers that do that. And so they keep the cells in free fall, and they basically subtracting gravitational forces from the cells and organoids. You can't put a whole organism in there. These are very small instruments that will take cells or organoids.
Which is quite an interesting thing in itself.
Today we can basically make
mini brains and mini hearts.
mini brains, 1 or 2 mini hearts. Yeah, we call them organoids, right? And then we put them in microgravity, and you can accelerate the aging in 24 hours. You have uh, a vast majority of aging features that get accelerated significantly by about 5 to 7 years.
24
So it's
hours.
pretty dramatic. And you so imagine they develop diseases until they develop. So brain organoids develop
Wow.
synucleinopathy. This is Parkinson's disease. Cardiac organoids have cardiomyopathies. When you look at gene expression and when you look at functional behaviors, or we also have immune organoids that come from tonsillectomies,
Tonsils.
from tonsil material, and we also expose them to simulated microgravity, and that way you can start to age those, mini organs and study the mechanisms of aging, but also screen
Wow.
for protective agents. So imagine you could do this in 24 hours, right? So the ability to screen hundreds of different potentially anti-aging molecules in 24 hours gives us this very unique window to that aging and to possibilities,
Wow,
therapeutic
my mind, my
possibilities.
mind is just blown when I heard this the first time. I was just like, for a start, I was like, how did you get from immunology to space travel? For starters, because I was like, that's really left field. And then I was like, now I get it, because it's an accelerated aging model that you can speed up the process of being, and you're perhaps going to help astronauts live healthier, longer lives, because these guys and girls are pushing their bodies to the absolute limits, coming back with age-related diseases for 1 year in space. I think it was 30 years of age advancement that they're coming home with, and thank goodness they do rejuvenate to a large degree when they come home with many of the things, but still they are aging themselves by doing that work. Yeah.
There are, depending on the endpoint, some hard endpoints may
Wow.
take many years to go back to baseline. So definitely it is an area of high interest to us. In fact, this all started from a grant that I had from NASA, from the Human Research Program, to protect astronauts. And so that's definitely on the table. But we are mostly pivoting on that accelerated aging mechanism to cure aging on
Because
the
you
ground.
get to accelerate the testing and the seeing what causes the aging and what stops the aging of these organoids in microgravity, we live in 1G. We've evolved in 1G, so anything that is above 1G or below 1G seems to cause accelerated aging. So not just microgravity, but if we were to go perhaps into 2G, 3G, whatever, we'd have a similar sort of problem. Is that right?
That's exactly right. So as I mentioned, I think anything that you experience that is new to you as a species will trigger inflammation. We talked about different exposome factors. Certainly, if you have evolved in 1G for the beginning of life on Earth, and you're deviating from that, either hypergravity or hypogravity or microgravity, you will suffer from an accelerated aging response. So I really see this as an opportunity for us to advance the field of protective geroprotectors, we call them, to target aging and age-related disease, ameliorate different maybe severity of disease, and also largely
this is
in the
absolutely
preventative side of
fascinating.
things.
And Dr. David, I know you've got to wrap up shortly because you've got to get to another appointment. But I could sit and talk to you for days, obviously, because I'm just absolutely fascinated by this work. And I think we've covered a lot of things. I think I'd love to have you back on when you have more research to share and to further this and to be involved with the iAge. Like I want to try that myself. I want to maybe do something with that. Yeah, let's do it. Let's make that happen. I'll
Let's do
reach
it.
out to Heather and we'll talk offline. But I think this is pretty pivotal, and it's quite a shift. And I'm quite excited about all of this because I really think that this is going to have a massive impact on humanity's health in the future. And, you know, give it another 4 or 5 years when this is fully matured and ripened, and things that you can do are going to be absolutely exponential. And I just want to thank you for such dedication. It's just absolutely amazing. And your dad was right. You have a bigger impact on a worldwide scale doing the work that you do. So it's been absolutely wonderful. Thank you so much, Dr. David. I really appreciate you.